we will be talking about immunity. First, our study tip is, I've said this before, if you're running a race, you practice, right? You train, and then you rest the day of. Well, now let's focus on the training part. So if you write on an exam, you should practice by writing, right? So you should be taking time to type out explanations of all of the main concepts from each chapter. You should also do this for lab, especially since now you know our format for online courses will be asking you one of the activities from each PhysioX, and you don't know which one. So write it out for each of them. And then when it comes time for the test, you'll have all of that material that you can copy and paste, or at least reference, okay? So practice writing complete sentences and explaining all of the concepts. Okay, so chapter 15, the immune system, yay! So we're going to talk about what is immunity. The two branches of immunity are innate and adaptive. So we're going to spend this first part of the lecture just on innate immunity. And then we will spend the second half of the lecture on adaptive immunity and a little bit on HIV, tumors, and allergies or diseases caused by the immune system, or rather when the immune system misbehaves. Okay. So today is all innate, remember. Okay, so immunity. Notice this little picture, just some bacterial cells are the long cylindrical guys, and then the spiky guys are viruses. You probably already know this. Okay, so all the structures and processes that provide defense against potential pathogens. So these guys, right? So a pathogen is any organism or agent that can cause disease. And so organism would include things like bacteria, protozoa, fungi, right? These are all living organisms. Viruses, however, are protein and nucleic acid. They're not living. Okay. Now, we tend to get, not me, but most people tend to be pretty freaked out, disgusted, scared of bacteria. But most bacteria is good. And whether that's good bacteria that's on your skin or in your gut or the environment, there are a lot of really important things that bacteria do for us. Okay, so things like decomposing. If we didn't have bacteria, the surface of the earth would be covered in feet, miles maybe, of dead tissue from, all the plants, all the animals, everything that's ever been alive that dies. Right, we need that to be decomposed, and we need it to be broken down, right, broken down into the individual molecules and returned to the soil and ready to be utilized for more organisms, right? We also have something called nitrogen fixers. These are bacteria, primarily bacteria, that live in the soil, and they're able to take nitrogen out of the atmosphere, which makes up about 70% of our air is nitrogen. And plants have to have nitrogen to survive, but plants are not able to take nitrogen out of the air. But these nitrogen fixers, bacteria and other microbes that live in the soil, are able to actually take the nitrogen out of the air and what we call fix or make it a part of the soil, and then plants can use that nitrogen and grow. We also have symbiotic relationships, which are where bacteria or other microbes live with another organism, and without them they wouldn't be able to grow. And so we actually see this a lot with plant roots as well, where the bacteria live in the root in specialized compartments of roots, and they perform very specialized, important jobs that allow the plant to thrive. Some plants will die if you were to remove their symbionts. We have other examples of symbiotic relationships. You've probably seen or heard of lichens if you took general bio, and that's a fungi and a bacteria that live together, and they can't live together. I mean, they cannot live apart from each other. They have to be together. And they're usually the first organisms to grow in an area. So let's take like an area that's been burned and everything's dead. Usually lichens will be the first organisms to start growing and getting that, basically recreating an ecosystem, right? Because once you have some life, you're going to get more and more other types of life that will be able to grow. Okay, so now you know microbes, super important, super beneficial. It's less than 1% of microbes are actually pathogenic or disease causing, right? Oh, and also food. I didn't even put food production on there, but we utilize bacteria for lots of different, and yeast for lots of different food production. And we can also utilize these microbes to make or synthesize molecules for us as well. So there's a lot of industrial applications. All right, super cool. I hope you, when you take microbiology, I hope you really enjoy it because it's such a fun topic. All right, so types of innate immunity. And innate immunity means non-specific. When we say non-specific, we mean you're not identifying the type of pathogen. You're just having an automatic response to any type of pathogen. So it's non-specific. You're not identifying the type of organism. So you wouldn't say like Staphylococcus aureus, which you guys know is like a staph infection, right? That's specific. So when you're talking about specific organisms, then you're talking about adaptive immunity. So right now, in this lecture, we're going to focus on innate immunity, non-specific. Okay, there's 7 major categories of innate immunity. These include our physical barriers. Then we have phagocytes or phagocytic white blood cells. Then we'll look at fever. We'll look at these special molecules called interferons, and inflammation, and then natural killer cells and the complement system. Okay, these are a lot of big topics that we could spend a lot more time on if we had the time. So we're going to get a brief overview. Recall that leukocytes are white blood cells and lymphocytes are T and B cells. We will not be looking at lymphocytes in part 1. We'll be looking at those in part 2. So today, leukocytes are white blood cells, and we'll be looking at, specifically, we'll be looking at phagocytic cells. All right, so first, let's unwrap what we mean by innate immunity a little bit more. So when we're talking about innate, as I said, it's non-specific, so there's no antibodies. Antibodies are super specific. So we cannot be talking about antibodies if we're talking about innate immunity. Okay, so no antibodies made or used in innate immunity. These are just automatic responses to any invader, any potential pathogen. Okay, the genes for innate immunity are inherited, passed down, right, from parent to offspring. Now, our body can recognize the difference between our own self, right, and anything that's not part of our self is considered a foreign antigen, and we detect that using what we call toll-like receptors. So this TLR that you see written here, T for toll, L for like, and R for receptor. So we have receptors on our cells, within our cells and on our white blood cells as well. Okay, and so we have these receptors that will bind to PAMPs. So what are PAMPs? Are pathogen-associated molecular patterns. Pathogen-associated molecular patterns. This refers to pretty much any part of a pathogen. This could be parts of its cell wall. This could be parts of its DNA, right? Pretty much every part of its cell, or if it's a virus, any of its proteins, all the pathogen parts are going to be unique to pathogens and different from ourselves. And so we will use our receptors, toll-like receptors, to recognize PAMPs, pathogen-associated molecular patterns. Okay, so some other examples of PAMPs would be like flagella of bacteria, right? It could be fungal cell walls, bacterial cell walls, any of these molecules of pathogens that are unique to pathogens, different from ourselves. Okay. And then we're going to see this come up again when our TLRs bind to a PAMP. It's going to trigger a response, and that's like the alarm system. So you can think of toll-like receptors of our receptors, right? Toll-like receptors are kind of like security cameras and other things that will be alarms will be triggered if PAMPs come into contact with TLRs. Okay, so we'll see that again in a minute. Okay, so we're going to start with the 7 categories of innate immunity. So physical barriers. This involves your skin, which we'll talk about on its own slide next. Sweat, tears, saliva, mucus. These are all liquids. You get physical removal, right? Microbes get trapped in the fluid and washed away, right? Same idea with mucus. So your mucous membranes, right? Your eyes, your nose, your mouth. You also have your intestines are mucous membranes as well. So the idea is to constantly have fluid or mucus going over the surface to collect and remove any pathogens and debris as well. Okay, so ciliary escalator, we'll take a look at that on its own slide as well, but this is in the lungs, right? In the lower respiratory system is lined with cilia, and the cilia will have mucus on top of them that they propel constantly up and out of the lungs. And so any mucus or other particles that get trapped, right, just from breathing your air constantly, you have particles that get trapped in your mucus. This is one of the reasons why it's so important to exercise and not to smoke. Okay, we'll come back to that. Urine also is a mechanical, physical removal, right? Just washing out bacteria. Stomach acid is unique in that it's not about physical removal at this point. Stomach acid's all about the pH, right? It's super acidic, and most anything, right? And I did say most anything will be killed or inactivated by your stomach acid. But you know that you do sometimes still get sick, right? Like when you have diarrhea or vomiting, right? Those are pathogens that were able to survive the stomach acid and give you a digestive tract infection. And then, of course, vomiting is also a response to flush out and remove whatever is in there. Okay, so in addition to this physical removal, we also have special molecules called antimicrobial peptides that can help attack pathogens. I already mentioned the pH of stomach acid. This also applies to you have a particular pH of your skin that is acidic. And also vaginal secretions are pretty acidic as well, and that's also to help avoid bacterial growth, right? Um, yep, I think that's everything I want. Oh, lysozyme. So Lysozyme. Lysozyme. It's an enzyme that lyses open bacterial cells. Okay, you have lysozyme in your tears, in your saliva, even in your urine, and your sweat, your perspiration. Okay, all of these, in addition to the physical removal, also try to attack any bacteria that are present with these lysozymes, right, to directly kill. So that's a really important component as well. Okay, so skin. Skin is super cool, right? You have layers, as you know, and so new skin cells are constantly replacing old ones. You're constantly shedding your skin. Okay, so the skin you see right now, look at your hands, right? The skin you see right now, it's gone, completely replaced in a month. Okay, so in a sense, you're a new person every month. Epidermis, the top 18 to 23 layers. So look up here, this dark region. Okay, so the top 18 to 23 layers are made of dead keratinized cells. So as these cells travel upward, they start to produce keratin, which is a water resistant substance. Your cells flatten out, they become very hard, and they are actually also, they die once they finish producing their keratin. And that's the very outer layers, or this very dark region, okay, the top layers of your skin. Okay, and those this serves several functions, right? So it's very dry, and bacteria and fungi, they need moisture if they're going to colonize a region. But even those that are able to survive with very little moisture, you're shedding your skin, right, constantly. So having the dead top layers allow you to shed, and a lot of organisms will be shed with it. Anything that started to colonize typically will be shed. Okay, so these are physical factors, how your skin is a physical barrier, mainly due to the dryness of the top layers of the epidermis, which is due to your keratin, and the continual shedding, right? Those are the two main properties of the skin that act as a protection against pathogens. Okay, now the dermis are the lower levels of your skin. Skin cells start out plump, right? So you can see here that they actually look round and plump like normal cells that you expect to see, and then they become flat, and it's hard to even distinguish them because this picture is so small. But they just become flatter and flatter until they're dead at the top with all their keratin. Okay, so dermis, skin cells start out plump. Over time, they move up to the top and flatten out and become part of the epidermis. Okay, so you have epidermis, dermis, and then we're not even talking about the bottom, the other parts, right? We're just focused on how skin acts as a physical barrier to protect you from infection. But I do want to just say some really cool facts about skin. In just 1 inch of your skin, you have approximately 19 million skin cells. Okay, first of all, that's incredible. About 650 sweat glands, 20 blood vessels, and 60,000 melanocytes, the things that make melanin or skin color, and about 1,000 or so nerve endings in just 1 inch of skin. So our skin is really incredible. All right, next, I told you we'd talk a little bit about the ciliary escalator, right? So if you were to look at the lower respiratory system, you would see these elongated epithelial cells, and then in between, scattered throughout, you have these specialized goblet cells. Now, the endothelial cells have cilia at the end. So you see all the hair-like projections, and that line, that's your whole lower respiratory system, okay? And so here you can see a little bit better these cilia, and then every so often you get a goblet cell, and the goblet cell secretes the mucus. And then here demonstrated is a trapped particle, right? This could be pollen, this could be anything, virus, bacteria. It gets trapped in the mucus, and because the cilia constantly move, they make a wave-like motion to propel that mucus up and out of your lungs. I highly suggest you watch this video. I think you only need to watch the first 3.5 minutes. And then it starts going into like research, and it's too, way more detailed than you need. But the first 3 minutes or so really demonstrate this, how this works. And it shows how incredible this system is. It's so fascinating to see how fast cilia move and what that looks like, and how it actually propels this mucus to protect us. So there you go, ciliary escalator. It's the cilia propelling mucus up and out. Okay, that's going to go up and out of your lower respiratory system, and it's going to dump it right into your throat, and you're going to swallow all of that. You make something like 1 liter of mucus every day. It's incredible. So stay hydrated. And exercise, right? Sneezing and coughing speed up the activity of cilia, but so does any accelerated breathing. So exercise that gets your heart rate up is going to get your breathing rate up, and then your cilia will work faster, and you'll clear out your lungs. And this is a really good way to stay healthy, right? And prevent respiratory infections such as COVID-19. Okay, innate immunity. Continued. Second of 7, right? So phagocytes. Phagocytes are a major component of our innate immunity. They go around and eat debris and look for invaders, okay? Neutrophils are the most abundant of all phagocytic cells. They are also, because they're so abundant, they're typically the first ones on the scene if you have an injury or any type of pathogen, or any type of infection that's starting. Neutrophils are going to get there first. Okay, so most abundant, first on the scene to fight an invader. Okay, then we have monocytes. Monocytes travel in your blood, and then when they become activated, they turn into macrophages, which macrophages will be active in the tissue to, again, to help clear whatever potential infection you have, or actual, right? Hopefully everything's getting there before it's a full-blown infection. You also have something called dendritic cells. Dendritic cells are a very important middleman between innate and adaptive immunity. So dendritic cells, they're able to take up a pathogen and digest it and display antigens from that pathogen. And so for this reason, dendritic cells as well as macrophages are called antigen presenting cells. I want to clarify that neutrophils are not antigen presenting cells, which we abbreviate APC. Neutrophils, they're phagocytic, but they are not antigen presenting cells. Okay, but macrophages and dendritic cells both are phagocytes that can also display antigen. And why would you do this? This is to be able to take this information in a safe way, right? You kill the pathogen, but then you display its surface proteins, its components that we call antigens. You display that to the adaptive immune system so that it can get to work and create antibodies and everything else that it does to then get an even stronger immune response that will also lead to a permanent immunity. So we do have a relationship, right, where innate immunity talks to and interacts with adaptive. And dendritic cells are the best example of this interaction, right? So they're the middleman. They're going around eating anything, right? So it's non-specific, but then they will go and talk to your T and B cells and let them know, like, hey guys, we have a situation. I found a pathogen. Let's get to work. Okay, so dendritic cells are antigen presenting cells. Their main function is to process antigen from a pathogen and present it on the cell surface, its own surface, and then present that to T cells of the immune system. So in this way, they act as messengers between innate and adaptive immune systems. Dendritic cells are present in tissues. Particularly, these tissues are the ones that are coming in contact with the external environment. So things like your skin, your nose, your lungs, your stomach, your intestines, right? So you're going to have a lot of dendritic cells in all of those areas. And then that gets us to the next category, because we're talking about dendritic cells, so I'm just going to skip over this for a moment and finish up our talk about dendritic cells, and then I'll come back, okay? So dendritic cells, I mentioned, can be present in the skin, the nose, the lungs, the stomach, the intestines. We also have specialized dendritic cells, organ-specific dendritic cells. So in the skin or the epidermis, we have the Langerhans. In the liver, we have Kupffer cells. And then in the brain, you have microglia. That these cells are always in place in these organs to also enhance our ability to detect any invader, right? We really, really want to protect ourselves from pathogens. And our body does an excellent job of this. You constantly get rid of pathogens every single day in multiple places, and you never know, okay? It's only when those pathogens start to colonize an area, right? They start to grow in the amount of cells, then you get enough of an immune response to have symptoms, okay? All right, so let's go back to our macrophages and dendritic cells up here. Okay, so if they do, so remember, normally they're just clearing debris. Cells die, break down, right? But if a TLR, toll-like receptor, does get triggered, then the macrophage or even the dendritic cell is going to secrete these pro-inflammatory cytokines called chemokines. Okay, so chemokines, you would know, oh, that's the specialized group of cytokines that promote inflammation and recruit local phagocytes into the tissue where the problem is, okay? And so we're going to talk a lot about this whole response right now. Okay. I just want to make sure I've cleared everything else. The only thing I didn't specify here, and again, because we're talking about innate immunity, I did just explain what antigen-presenting cells are and how your macrophages and your dendritic cells can present antigen, so they're called APCs, antigen-presenting cells. We also have one type of APC for the adaptive immune system, and those are your B cells. So a lot of information for you to write down, right? from this slide in particular. Okay, so here's a little summary of the types of phagocytic cells and where they're found in the body. So neutrophils travel throughout your blood, and they're constantly moving out of the blood into the tissues. Okay, and they're the most numerous. First responders to any infection or damage. Okay, monocytes also travel through the blood. These are like immature inactive cells, and then if they become triggered or activated, they will turn into macrophages. Okay, and in that case, once they're a macrophage, then they're going to be performing phagocytosis. Okay, you can find these everywhere in the body, and that makes sense because the monocytes are traveling through the blood, so they're going to have access to the whole body. So all of your tissues, including your spleen, your lymph nodes, bone marrow, right, everywhere, you're going to have macrophages. Then specialized dendritic cells, right, we talked about in the liver, the Kupffer cells, the lungs. I actually didn't talk about these, but this is super important. We have, remember, your alveoli are the individual little, look like little sacs or little balloons. They are just a single layer, and that's where gas exchange occurs. So if you think about inhaling and that air that you inhale going all the way to the very end through the whole branching tree to the very end of the lungs is the alveoli, where gas exchange occurs. And it's super important that we keep that clean. And so we have these alveolar macrophages that constantly clear any debris, and that would include getting any pathogens that may have traveled that far into the lungs. Okay, so alveolar macrophages, super important. And then the microglia, right? We have that blood-brain barrier. We don't allow much of anything through to our central nervous system, but we do have specialized dendritic cells called microglia that protect our central nervous system. Okay, so now we're going to talk more about these phagocytes and how they respond to if you cut yourself. Okay, so first we need to understand, what are phagocytes? Phagocytes are white blood cells that perform phagocytosis. We talked about the different types just now, right? These are the list of phagocytic cells. So what is phagocytosis? Phagocytosis is when one of those specialized cells, right, a neutrophil, a monocyte, whatever, not a monocyte would have to mature into a macrophage. Okay, so let's say this is a macrophage right here, and it is grabbing bacteria, and it's going to take that bacteria inside. It's going to digest it. It's going to get rid of it. It's going to kill it. Okay, and so the steps of phagocytosis would be, first have to extend a part of the cell that we call a pseudopod. We need to extend out and actually go towards the pathogen bacteria. Okay, so chemotaxis is going towards a stimulus such as bacteria, and then you need to grab onto it or adhere. So right here, this first step, chemotaxis and adherence of phagocyte to the microbe. And you can see here, we've enlarged what's happening is there's that toll-like receptor on the phagocyte, and it's binding to a PAMP, a pathogen-associated molecular pattern. In this case, the PAMP is the cell wall of this bacteria. And so it grabs on to the bacteria, and then it's going to pull it inside of itself. So ingestion of microbe by the phagocyte. And we don't want it to just be inside and have free reign to do whatever it wants. So we're going to put it inside a protective vesicle called a phagosome, which phago for phagocyte, right? Vesicle, some. Okay, so formation of the phagosome you have your bacteria stuck inside this vesicle. The next thing that's going to happen is you have a lysosome. A lysosome has digestive enzymes like lysozyme, and that's going to then fuse with your phagosome so that the digestive enzymes can digest the bacteria. So fusion of the phagosome with the lysosome to form what we call the phagolysosome. Okay, and then digestion occurs because now you've released those enzymes into the compartment, right? You've made one big compartment. And so you digest this microbe, and then you just have all the waste from that digestion, and then you can release it. Okay, so discharge of waste materials is super, super small, and this will just get carried away in your like your interstitial fluid. That will then become part of the lymph or part of or directly into the back into the blood, and then like your kidneys will filter that out, and then you're done. Okay, so phagocytosis, chemotaxis moving towards the bacteria, adherence grabbing onto it, ingestion to form your phagosome, digestion when your lysosome fuses with your phagosome, then you digest it, and then discharge of waste. Okay. Now, let's assume that we have an injury. We've accidentally, maybe we're cooking, right? And we accidentally cut ourselves. So everything, the air is full of bacteria and fungi and things. And so all of anything in your house and anything you ever come into contact with is going to be covered in bacteria that you cannot see, right? So I know some people look at this image and they think, ew, why would you be using a dirty knife? Well, this could be a clean knife that you washed and dried, and it's still going to have bacteria on it, okay? So you accidentally cut yourself, it has bacteria on it. Now that bacteria has been allowed to go through your protective barrier and into your epidermis and even into your dermis a little bit, okay? Your body is going to respond, all of those cells that you just tore open and damaged, they're going to release chemicals, histamine, kinins, prostaglandins, and other cytokines, right, are all going to be released by this damaged tissue. And that's going to cause this is very, very fast. You're going to vasodilate. It's going to swell, right? You're going to get extra blood flow so that you get a very strong, immediate response. At the same time, right, you're going to be forming a blood clot to stop the bleeding, okay? And then your body will work to heal that. And at the same time, your body has to work to make sure that you don't have an infection, okay? So now an abscess starts to form here where all these chemicals, cytokines, and whatnot have been released. And also that's where all the bacteria are. So these tiny little rod-like objects are the bacteria, right? They look just like what we saw here, the bacteria. And then all these blue dots are the chemical messengers from the damaged tissue. And then, of course, these are some capillaries, right? And notice that from before and after, we have a lot more blood flow. We have lots of vasodilation. So increased blood flow. And then also all of these chemical messengers and all this extra blood flow is going to allow for extra permeability of the blood vessels. And that's how you're going to get your white blood cells, particularly your phagocytes, are going to enter the abscess, right? So they're going to travel through the blood and exit the capillaries and take care of these invaders, okay? And so this is a very general, I'm showing you this process first as a summary or a very general explanation, and then we're going to look in a little more detail. Okay, so going from here, right, you have bacteria in this abscess. Now we need to get the white blood cells, the monocytes, so that they can become macrophages, right? So notice it's a monocyte here, but now it's a macrophage in the tissue. You have your neutrophils as well, and lots of neutrophils, right? Look how many, we have so many more neutrophils than anything else. Okay, so these neutrophils, as well as your monocytes, are going to, what we call margination, they're going to get near the wall of the capillary, and then they're going to stick to it and squeeze through. Okay, so phagocytes squeeze between the endothelial cells, and again, that could be your neutrophil, it could be your monocyte, any phagocytic cell. Okay, and then they go into the tissue, and then they perform phagocytosis. They eat up the bacteria. Okay, your blood itself is not exiting the capillary. Okay, or not any more so than it normally would. Your red blood cells never leave, just fluid, right? We talked about that in the last chapter. Okay, so then once all that's done, you've gotten rid of all the bacteria, all of your chemical messengers have, right, you don't have any more chemical messengers being produced, so everything calms down. You stop having the vasodilation, you stop having the, what we call extravasation or diapetesis is the process of these white blood cells going into the tissue. All of that calms down and stops, and then you're just left with the wound healing, right? So you get that scab formation, and then over time you regenerate your epidermis, the blood clot and scab go away, and you have completely healed skin, right? So that's the overall process. Now let's focus in on how exactly did the white blood cells get into the tissue. Okay, so chemokines attract first the resident neutrophils and monocytes that are already nearby, and of course that's through chemotaxis, right? And they're going to perform phagocytosis just like we talked about with any of the pathogens present. But if there's enough bacteria present, they might not be able to handle that on their own, and they might need to recruit more phagocytes to the area. And so they would release more of these chemokines, and you would get new phagocytes from the blood to come to that area. And that process, like I mentioned, is extravasation, which, if you break that word down, it looks like vasation, vessel, right? And then extra, to go outside of. So that can help you remember, like, extravasation is the process of these phagocytes to be able to leave the vessel, the capillary. Okay. Another word that's used for this process is diapetesis. Please watch this video. It's very short and it goes over the steps. It's really good. Okay, so I'll go over it with a picture, but please watch the video. Okay, so how phagocytes are able to move from the blood to the site of the infection. They're recruited. You get more of them, both because of your vasodilation, but also because they're attracted, right? Chemotaxis, they're attracted to the chemokines that have been released. Chemokines have been released from damaged tissue, and they're also released from the resident phagocytes that are saying, hey, we need help, we need backup, right? Okay, so then your neutrophils and your monocytes are going to get near the vessel wall, and then you're going to have these weak bonds that form again between the neutrophil and the wall of the capillary. It's not shown here, but there are weak bonds made, and because blood flow is, we talked about all that pressure, there's all that pressure, there's all that blood flow, so these weak bonds are not strong enough to hold the neutrophil in place, so you get what we just call rolling. That's exactly what happens, is that this neutrophil is going to roll along the surface of the wall until strong bonds are made. And so that's captured. And you see it here. A strong bond is made. So now it's captured. It adheres to the surface. So now you have this direct talking is able to happen between this neutrophil and this endothelial cell. And that's going to allow the endothelial cells to loosen their tight junctions. It's also going to allow the neutrophil to do this spreading out action. And so together, the communication of loosening the capillary and spreading out of the white blood cell, it's able to then squeeze through and get into the tissue. Okay, so this process can be set in motion in response to damaged tissue, having a lot of bacteria in an area, such as an abscess, which could happen from an injury like we saw with the knife, but it could also happen just from bacteria getting through like a hair follicle, right? Something like that. Or even your normal flora that for some reason overgrow. Okay, so lots of reasons why you might need to have white blood cells come and clean up bacteria from a tissue. Okay, so I know that was a lot. So here are the steps. And remember, you could call it diapetesis or extravasation. Response to chemokines in an affected area. Endothelial cells of the capillary express these adhesion molecules called selectins. And this is going to allow for the rolling adhesion, right? Remember, weak bonds between the phagocyte surface and the endothelial cells form. So the endothelial cells have these projections, these molecules that stick out from their surface called selectins. Phagocytes have their own surface molecule that will project out, and they'll grab onto each other, but they don't grab on very tightly. And so the force of the blood flow is going to cause the neutrophil to roll against the surface of the capillary. But then you will end up getting the formation of integrins on the phagocyte. These are specialized binding molecules, and they're going to then bind to endothelial cells, specialized molecule called ICAM1. So because of the weak bonds, you've stimulated these other molecules to grab onto each other, and that's what we call capture, and that leads to very tight binding, and then you have your adhesion, your activation, and your spreading. So now you get all this cell-to-cell talk, right? Essentially the neutrophil is telling the capillary, like, hey, let me through. And so then the tight junctions between the endothelial cells will loosen, the phagocyte will flatten out, and then it will squeeze through. So diapetesis is the actual squeezing through, but we also refer to the whole process as diapetesis too, which is a little confusing, I know. All right, so this was a lot of information. Please take a lot of time, watch the video, look at the slides, really take your time to know all this. It's a lot of information. Okay, next, fever. Fever is actually really good. It's a general response to an infection. We have special chemicals called pyrogens that actually tell your hypothalamus to change the set point to a higher set point. And so then your body thinks it's not warm enough, because now instead of being told to keep your body at 37 Celsius, it might be saying 38 Celsius. And so you need to quickly warm up. And so to do that, your skeletal muscles are going to shiver. You're going to have extra heat production from your brown adipose tissue as well. Your heart rate's going to have to increase. You're going to have increased metabolism to get all this extra heat. Okay. At the same time, because your body thinks it's really cold, it's going to vasoconstrict especially like your feet and your hands. Okay, typically what during this stage, a person feels like they're freezing cold, they're shivering, they're trying to warm up, they might pile blankets on top of themselves, right? In actuality, if you were to measure their temperature, it'd be more higher temperature than normal, right? Benefits of a mild to moderate fever: you get increased proliferation of your T cells, particularly T cells and B cells, to be able to make antibodies. But all of your immune system will be stimulated to be more active. Okay. Now, here's a better layout. So advantages: the increased temperature from the fever will help to kill, and if it doesn't kill the pathogen, it will just at least slow it down, so that it's you're slowing down this infection. And then in the meanwhile, you're increasing the activity of your immune system so it can get rid of that infection. This also is going to increase the rate of tissue repair. So any damage that may have been, any damage that may result from the infection or the fighting of the infection will then be repaired. And then you also have interferon production. We're going to talk about interferon next on the next slide, but this is a special molecule to help fight infection as well. Okay, disadvantages: fever can be dangerous. So it does cause a rapid heart rate, which for a healthy, normal individual is not a problem. But if you have a heart condition or other certain conditions, then it might be more dangerous. And you would have to monitor that more closely. You can become dehydrated easily. You have all this extra activity happening. You have a higher temperature, right? So this is one of the reasons you're always told to drink lots of fluids when you're sick, so that you don't become dehydrated. If you become too dehydrated, you can die. And people die from dehydration all the time, especially in other countries. So it is important. Of course, if your fever gets way too high, it can cause you to hallucinate or be delirious. It can even start to cause actual brain damage from denaturing proteins, right? And then, of course, if enough of that happens, that can lead to death. So death can happen from brain damage, dehydration, heart problems, right? So this is why we take our temperature and we monitor it to make sure it doesn't get too high. So you really should let your fever help you. Don't take fever reducers until it starts to get too high or it's been more than a few days, okay? So when do you treat a fever or see a doctor? Well, for babies, they are much more sensitive. So 100.4 Fahrenheit is a high fever for a baby, okay? If the baby is 3 months or older, it can handle more at that point, 102, okay? Of course, at 3 months exactly, if it was getting to 100.4, I would still contact the doctor and see exactly what they say. A lot of times you can call in, right, and speak to the doctor's assistant or nurse and see what they have to say. For adults, so, teenagers and up, 103 is getting a high, right? That's a high fever. So never ever let a fever go above 104. So once your fever's at 103, you might want to call the doctor and see about taking some Tylenol or fever reducer, okay? All right, innate immunity continued. I told you we would talk about interferon. So here, advantages: increased interferon production. So what is interferon? It's a polypeptide. It's released from infected cells. So when a cell detects foreign nucleic acid, like DNA, but it could also be RNA, it's in the cytoplasm. It's like, well, what is this? We have an invader. We have nucleic acid in our cytoplasm. It's not ours, right? Because our nucleic acid would be secure in the nucleus. Okay, so for all sorts of reasons, we're able to detect foreign nucleic acid that's in our cytoplasm, and we're going to respond. And this is basically like, it's too late for me, but save yourself kind of response. So this interferon is going to talk to nearby cells and help them to lock down so that they don't get infected. We have alpha interferon, we have beta interferon. These, again, I said interferons will help trigger the nearby cells to produce virus blocking enzymes. These enzymes block the virus from being able to replicate inside of the cell, so you stop the infection. But that only works for the cells that receive the interferon. So something farther away is still going to be susceptible. Okay, but this does really help. And in fact, interferon has been given as like a supportive therapy for people who are fighting viral infections that they're in critical condition, right? We also have some lymphocytes and natural killer cells that release something called gamma interferon. And this is useful to fight infections, certain infections, and also cancer. Okay, so interferon can be a very powerful tool. All right, that's enough detail there. Next, local inflammation. Well, we already talked about that quite a bit, right? So when you get that tissue damage or foreign particle detected, those resident phagocytes and mast cells release your chemokines, your histamine, and that gets extra blood flow, so you get that vasodilation in that local area, right? You're also going to increase permeability so that diapetesis can occur. So recall those steps that we went over. So this localized vasodilation, right, is to increase the blood flow to the area so that you can get more of the phagocytes to perform diapetesis and get into the tissue and clear out whatever's going on. So symptoms in the local area is going to be swollen, red, warm, painful usually. You might even get some pus production, and the white part of pus are white blood cells. If it's other color, then that can be from the bacteria itself. So here's a link that you can watch to help also review inflammatory response. You may have been wondering, well, what's a mast cell? She never talked about that before. A mast cell is a cell filled with granules, which release histamine, and that increases vasodilation. Natural killer cells, non-specific, right, because we're still talking about innate immunity. So natural killer cells have non-specifically destroy virus-infected cells or cancer cells. And this would also apply to a transplant that was not done correctly. So if you have a mismatched transplant, then your natural killer cells are going to attack. Okay, so cells, your own cells, if they become infected or if they start to become cancerous, they change, and they change specifically what we call MHC1 self-tag. They will change the way that it looks, and so then your natural killer cell will kill it. If it's a mismatched transplant, it's not going to have your own self tag. It's going to have some other MHC tag, so then that will also be attacked. Okay, so our cells mutate all the time and become cancerous or precancerous, but our natural killer cells destroy them before it becomes an issue. Okay, how do they do that? They will cause cell death. They will cause the cell to burst itself open. Okay, how does it do this? The natural killer cell has granules that contain perforin to punch holes in the wall. There's also some other chemicals that will tell the cell to chop up its own DNA to really make sure that you kill this cell. Okay, and that makes sense. You need to prevent cancer. You need to kill infected cells and get infections under control. Okay, so they were named natural killers because of the initial notion that they do not require activation to kill cells that are missing the self markers, the MHC1 tag. This role is especially important because harmful cells that are missing the MHC1 markers cannot be detected by any other type of cell in our body. So our natural killer cells are the only cells that can detect missing or mutated MHC1 tags. Okay, super important. All right, complement. Complement is not strictly part of the innate immune system. This is why I put it last. It's also kind of an intermediate between innate and adaptive, and you'll see why. So it complements the innate system, but it also complements the adaptive system. So it will also complement the action of antibodies, which would be specific adaptive immunity. Okay, activated by foreign antigens, which you can say carbohydrates or any other type of antigen on a pathogen. Okay, so we're not looking like at what type of pathogen it is. It's a pathogen. And the other way to activate the complement system is if you actually had antibodies on a pathogen. So 2 ways to activate complement. One through the non-specific and one through the specific pathway. Either way, once you activate complement, which is a protein always in your blood, and once it's activated, it splits, and it's going to continue to cause this cascade of all these proteins, and then those proteins can come together and build what we call MAC, or membrane attack complex. So all of these proteins will then embed into the foreign cell and punch a hole in it. So osmotic influx of water is going to cause the cell to lyse open. This is very similar to the perforins we were just talking about back here with natural killer cells releasing granules of perforin. That's going to punch holes and lyse the cell open. So when you create a weakness in the cell wall, it's going to give in to osmotic pressure and water is going to flow in and burst open the cell. Other complement functions, functions from activation of the complement system is increased phagocytosis and increased inflammation. These proteins, the complement proteins, once you've activated them, you get all sorts of this cascade of all these proteins. They can line or stick to your pathogen and help phagocytes to grab onto it easier. And they can also increase inflammation as well. Okay, so here you have, this would be the pathway that uses antibodies for activation. So this would be how it complements the adaptive immune system in this example, is that you have some antibodies that are bound to the surface of your bacteria, and then the complement protein that's always in circulation in the blood sticks to it, and that's going to cause this protein to split apart, and it's going to activate that cascade I was telling you about, and then that's going to have all those individual proteins embed into the bacterium and form this membrane attack complex, and that forms a small hole, and then the water rushes in and lyses it open. Okay, good job. I know we just covered an hour of information. Go take a break, review all these concepts, watch your video links, and then you can watch the next lecture. All right. How do I stop recording?